Hello, everybody. This is Nafrit Seko, and I welcome you on this platform where I would love to be joined by you in this journey to decipher the life layer by layer. This journey will cover all the structures and procedures in depth and detail which are keeping us alive and the life around us. I hope through this journey you will not only become more confident to score good in your exams and sail through all the competitive exams, but ultimately you will also become more aware of your surroundings and the life within you. So, without any further ado, we are gonna set on this long journey which will cover the topics of biochemistry, cellular biology, molecular biology, genetics, animal physiology, plant physiology, discussion about the diversity, everything related to life sciences needs to be discussed. And we are gonna discuss this layer by layer. So, we are gonna begin this journey with a very simple, very basic, and kind of cliche question. And I know that of you must be knowing the answer to this question. And that question is that what is the basic unit of life? That's correct. The basic unit of life is the cells. So cells are the basic unit of life. Now, why the cells? Why the cells are called the basic unit of life? The reason is that they are the smallest entity which can sustain itself, survive on its own, grow, and finally reproduce. So these are the criterias in which using which we have categorized whole of this world into categories, that is living and non-living. We have kept the viruses on the borderline because sometimes the viruses show the characteristics of those of living world, and sometimes they just behave like the non-living things. All the unicellular organisms, I would say, are made up of super confident cells, and those cells say that we don't need anybody else. We are good on our own, and we don't need any help. So they sustain uh, just independently, and they grow, and whenever they find themselves to be grown enough, they just divide, and that division is basically their way of reproducing. The one mother cell produces two daughter cells, and this is the way they reproduce. Now, again, in that living world, there are the cells which are not that confident. They find the need to be joined by others to survive. So all such cells just come together, cooperate with one another for the survival, do the division of labor, get specialized for the special functions, and all such cells form the multicellular organisms. So whether unicellular or multicellular, all the organisms are composed of basically the cells. That's why the cells are called the basic unit of life. Now, that's true, right? So whole of this life on this planet is made up of basically made up of cells, right? Then the level of complexity may increase, like in different organisms, like unicellular, the simplest ones fall in the phylum Protozoa. Then in phylum Coelenterata, there are just the sponges, right? Loose association of many cells. Then in or other organisms, there is just the complex association of these cells. So depending upon whatever is the complexity of the life, whether the life exists in a very simple form or in a very complex form, the basics remain the same, and the basis of that life is the cells. That's done. Now, there is further question. The further question is that of what these cells are made up of, right? So, like every other thing in this world, cells are also made up of atoms and molecules, right? Atoms trying to form molecules. Now, then, what is so special about the cells that they are able to form the life, right? Why everything is not able to form the life? Everything is made up of atoms and molecules. Then why the cells? Now, what happens that cells are made up of particular types of molecules. These specific molecules interact with one another, undergo conversions and interconversions in such a manner that wherever these processes are taking place, the space in which these processes are taking place, that space becomes self-sustaining entity, and that space we call as the cell, right? Or it is a 3D space, right? It's not 2D space. It is a 3D area where these conversions and interconversions take place, right? So it is basically a small space, or we can say a small room, where these chemical processes take place, and these are so specific that it leads to self-sustenance. And this is the way something, molecules are non-living, right? So non-living molecules are leading to the living cells. That's why biology or life sciences is nothing but the living chemistry, right? Basic unit of life is cell, but the basis of life is basically the living chemistry. So, therefore, if we really want to understand life, we need to start from this chemistry, this living chemistry. And whenever this living chemistry is called biochemistry. So, whenever we need to understand life, we need to start from biochemistry. So, we are going to start with biochemistry. Now, these molecules, which are like very, very specific, they go so many conversions and interconversions. These are called biomolecules. All of these are called biomolecules. Now, these biomolecules can be proteins, lipids, nucleic acids, vitamins, carbohydrates. All these biomolecules are basically the macromolecules, which means that they are very large molecules. And these large molecules are made up of the smaller subunits. Like, for example, if we want to discuss the smaller subunit for proteins are the amino acids. For lipids, these are basically fatty acids and the glycerol molecules. For nucleic acids, it's nucleosides. Vitamins, it's just it is everything like amino acids, lipids, fatty acids, nucleosides, like in vitamins, everything exists. And for carbohydrates, it is like monosaccharides, right? So all these biomolecules structurally are macromolecules, which means that they are quite large molecules. These large molecules are actually made up of smaller subunits. So now it is like box within box kind of situation. Life is basic unit of life cell. The basis of that cell are biomolecules. Now the basis of these biomolecules are basically their subunits, smaller subunits. So all of them are equally important: proteins, nucleic acids, vitamins, and carbohydrates. Everything is really important for our survival. But because the most abundant ones are the carbohydrates, so we are going to start the discussion with the structure of carbohydrates. Let's start with the very first topic, the carbohydrates. And in that, we need to know different types of carbohydrates. So the carbohydrates have been categorized into three categories. These three categories are monosaccharides, oligosaccharides, and polysaccharides. Now, the basic units of carbohydrates, no matter how large that carbohydrate is, the basic subunit of all the carbohydrates are the monosaccharides. Monosaccharides is just as if there is just one block over here. Just they are not square or rectangular, but it's just the representation. So monosaccharides, a single unit, right? Now oligosaccharides, these monosaccharides have the ability to form the bonds among themselves and to form the chains. These chains may be branched or unbranched. It can be anything, right? So if this chain is composed of 20 or less than 20 monosaccharide units, that carbohydrate is basically called oligosaccharide. Now, but if this chain, the number of monosaccharides in these branched or unbranched chains exceeds 20, it usually goes till hundreds, like 200, 400, 600. There are so many polysaccharides where the number reaches till hundreds. So they are such big molecules. They are and all such molecules are called polysaccharides. So this is the basic classification as well. And this is the basic structure of the carbohydrates. This gives us an idea of the basic structure as well, that larger carbohydrates are made up of smaller units, and those smaller independent units are basically the monosaccharides. When these smaller independent units form the bonds among themselves, they can form the chains, which can be branched and unbranched. Less number of monosaccharides in one chain, the carbohydrate will be known as oligosaccharide. More than 20 monosaccharides in a chain, branched or unbranched chain, then those will be called polysaccharides. So here the conclusion of this becomes that monosaccharides are the building blocks of carbohydrates. Now you must have noticed that there is a word root over here, which is saccharides, and prefixes are just added in front of those that word root, right? Saccharides is the word root, is the common thing. Now, saccharides actually means sugar, and it has been derived from the Greek word saccharon. But does that mean that all the carbohydrates are sugary or sweet? No, not all the carbohydrates are not sugary or sweet, but there are so many known carbohydrates which are actually sweet, and they are very common carbohydrates in our which we actually use in our daily life, and we are utilizing them in our bodies as well, right? So we are gonna discuss these things, but before that, we need to discuss very important thing about all the biomolecules. One thing which we have studied about all the biomolecules is that they are macromolecules. Each and every kind of biomolecule is made up of smaller subunits. Second important thing is that all these biomolecules are basically the carbon compounds. They are basically the carbon compounds, all of them, right? We will be studying the structure of proteins, lipids, nucleic acids, and we are doing the structure of carbohydrates. Like you will realize that, yeah, the most abundant element of chemistry, if there is any, in the living system or in the biomolecules, it is the carbon. So to understand life, we need to understand, first of all, this element, this atom of carbon, right? So carbon is denoted by the symbol C, right? It is the member of 14th group of the periodic table, right? So there is a periodic table in which all the elements, all the known elements, and some unknown as well, they have been just classified into groups, right, according to their properties. But also, in addition to that, according to the number of valence electrons they have. What are those valence electrons? You may not be knowing, but we will discuss over here. Don't worry. So carbon is represented by symbol C. It is the member of 14th group of the periodic table. Its atomic number is generally denoted by the symbol Z, right? So its atomic number is 6, right? Its atomic number is 6. So what does this number 6 means? Atomic number 6 means that one atom of carbon has 6 electrons and 6 protons within an atom. So, so it gives us the number of electrons and protons. So according to Bohr's atomic model, right, it is made up of, there is a nucleus in the center, and right? And in this nucleus, certainly we have the 6 protons, right? So our 6 protons equal to 6 are over here, right? 6 protons are there. Apart from protons, there are neutrons over there, which may be, depending upon which isotope of carbon it is, they may be 6, they may be 7. Apart from that, this is the central thing. This is the nucleus. Like this is it is in the center of the carbon. Around the nucleus, there are the energy shells, which are also called energy orbitals, right? And in these energy orbitals, the electrons are arranged. The electrons actually are accommodated in these energy orbitals according to a formula, which is 2n square 2 times 2 times n square. Now, what is n over here? N over here is that which number of shell we are actually, for which number of shell we are trying to find the maximum number of electrons which can be accommodated in that shell. So if this is the shell, this is the first shell over here, right? This is the first shell. So and for that, we will put in the formula the number 1, right? So it means in the first shell, only 2 electrons can be accommodated. So that there are the total 6 electrons over here, right? Out of these 6 electrons, 2 have been accommodated in the first shell. So we are left with 4 more. Now, in this, if we try to find the total number of electrons which can be accommodated in the second shell, according to the formula, we will put 2 over here, and we do it, then it becomes 2 times 4, which is 8. So according to this formula, in the second shell, 8 electrons can be accommodated, but we are left with just 4. We had total 6 electrons. Out of those 6 electrons, 2 have been accommodated in the first shell, and rest 4, we are left with only 4. So those 4 are accommodated within the second shell. Now, this arrangement of electrons in the energy shells, or we can say energy orbitals, gives us the electronic configuration of that atom. So that's why the electronic configuration of carbon is 2, 4. Right here, let me make the things clear. We are discussing whatever is important from the biological point of view or from the biochemistry point of view. This is not very deep chemistry, but the chemistry which we require for biochemistry, this much is enough. So just coming back to the topic, so our carbon has atomic number 6, which means it has 6 electrons, 6 protons. The 6 protons lie within the nucleus in the center, and the electrons are arranged in the energy shells according to the formula 2n square, right? And we find that the first shell is filled to its capacity, which is that there are maximum number of 2 electrons can be accommodated. So there are 2 electrons over there. In the second shell, its capacity is that of 8 electrons, but we are after giving 2 electrons to the first shell, we are just left with the 4 electrons. So those 4 electrons are arranged in the second shell, which gives us the electronic configuration of carbon to be 2 and 4. Now, after this, very important thing which we need to discuss is the noble gases. Now, why we need to discuss the noble gases? Noble gases, there are the noble gases in the 18th group of the periodic table. Now, why they are called, they are also called inert gases, right? Why they are called noble gases? Because they don't react. No matter how much the other elements of the periodic table provoke them, they just don't react. That's why they are called noble gases. Now, why they don't get, why they don't react? They don't react because they are satisfied. Okay, they are satisfied. Now they are satisfied about what? They are satisfied with their electronic configuration, with their electronic configuration. Now, what is so special about their electronic configuration? The special thing about their electronic configuration is that they are fully filled, right? Fully filled. If somehow the shell is not fully filled, then the subshells within those shells are fully filled. Argon is one of those cases. Anyway, we will go there. So this is, they have, because they have fully filled electronic configurations, the noble gases are satisfied, so therefore they don't react, and because they don't react, they are called noble gases. Let's do some examples over here, right? So the examples can be is that of most common is that of helium, which is like the first noble gas in the group 18th. It has the atomic number helium. Helium has the atomic number 2. There will be just one shell, and in that shell there will be just these 2 electrons can be accommodated, and that first shell will be fully filled, right? So it has the electronic configuration of 2, like one shell which is fully filled. So it is satisfied, it is a noble gas. Then the second noble gas is that is neon. Neon atomic number is 10, and its electronic configuration will be, of course, the first 2 electrons will go to shell number 1, right? We are left with 8 electrons, so the second group of 8 electrons will go to the shell number 2. Now, in this, as we have discussed in shell number 2, the maximum electrons which can be accommodated is 8 only. So again, there are 2 shells and both of them are fully filled. That's why neon is also very satisfied with its electronic configuration, and thus it doesn't react. And it's a noble gas. Then third case is that of argon, which is like its atomic number is 18. Then it is electronic configuration is gonna be 2, 8, right? 2, 8, 2, 2, 8, 10. Now, after that, we are left with 8 more electrons. So 8. Now, this is, we had to go to the third shell. Third shell means, let's put the formula over here. 3 square, right? Which means that it can accommodate 2 times 9, which means it can accommodate 18 electrons, but here there are just 8 electrons in this shell. It means, does that mean that argon is not a noble gas? No, it is still a noble gas. Then why it is a noble gas? It is a noble gas because, as I have discussed, that shells are not the absolute thing, right? Within these shells, there are the subshells. Now, over here, with 8 electrons in the third shell, the subshells, the available subshells are fully filled, basically. S and P subshells are fully filled, so they don't require, so that gives it enough satisfaction. So, why we are discussing noble gases over here? It's very important to discuss. If we are doing chemistry, it's very important to discuss the noble gases. Reason being that every other element of the periodic table in chemistry wants to have the electronic configuration just like the noble gases, right? They are tirelessly trying to become the noble gases. And in this pursuit to have the electronic configuration, which would just look like the noble gases, they just react with one another. Like each and every atom of each and every element, like apart from group 18th, all the elements till group 17, they just react, right? And their main aim is to have the electronic configuration just like that of either helium, neon, argon, krypton, radon, right? They just want to have the electronic configurations like that. Carbon is not different in any way. Carbon has, in the second shell, it has 4 electrons. So to have the nearest noble gas, so its atomic number is 6, and the nearest noble gas has the atomic number 10, right? It is nearest. So each and every element wants to have the electronic configuration of its nearest noble gas. Everyone wants to take the shortcut, right? Nobody wants to work really, really hard. So they find the nearest noble gas really look like in the terms of electronic configuration. So what novel over here, this carbon has just 4 electrons. Neon is the nearest one. Neon has the electronic configuration 2,8. Carbon has the electronic configuration of 2,4. To become 2,8, it needs 4 more electrons. Now, if carbon would have believed in giving the 4 electrons, then it could easily get the electronic configuration of that of helium. Good for it. But no, nobody wants to give 4 electrons. He would with the any element which is just in the center of the things, that element say, I will gain 4. Why I'm gonna give 4? But now to gain 4, any other element needs to give 4 electrons, right? No, but no other element is ready to give 4 electrons. That's why carbon generally does not form the bond which is based upon this give and take. One needs, one takes, other doesn't need, that gives, right? Now, this given the bond which is formed by one is giving, another one is accepting. This such bonds are called electrovalent bonds. But carbon is not forming these give and take bonds, and uh, thus it believes in the bonding which is based upon sharing, right? The bonding which is based upon sharing. So such bonds are called covalent bonds. Such bonds are called covalent bonds. So let's see how it works. All right. Now, what is there in the what happens in the bond of sharing, right? Sharing is like that they just crack a deal, right? So like this is the carbon atom. It may crack a deal with the oxygen atom, right? So what does the carbon atom say? That I need 4 electrons, and if we see oxygen, oxygen is atomic number 8. Atomic number 8, which makes its electronic configuration to be in the first shell 2 electrons, and 8 minus 2, and for the next shell we are left with just 6 electrons, right? So nearest noble gas is again neon for both of them with atomic number 10 and electronic configuration 8. So oxygen also wants to become 2,8. Their carbon also wants to become 2,8. Both of them wants to have the electronic configuration that of neon. Now, here I'm going to draw the Lewis structure. So what we do in the Lewis structure, we draw the valence electrons. Now, what are the valence electrons? Valence electrons are the electrons in the outermost shell of the atom. So here this shell, second shell is the outermost shell for carbon, right? So this is the valence shell. This is the valence shell. Now, valence shell. Now, all the electrons in this valence shell are basically the valence electrons. So the valence electrons you can see are just 4 in number. So around carbon, I will draw 4 electrons. 4 electrons, right? 4 electrons are drawn. Now, in the valence shell of oxygen, like the outermost shell of oxygen, which is this one, there are 6 electrons. So we will draw 6 electrons around oxygen. Around oxygen, 6 electrons will be. So what deal can be there? Now the deal is that whenever I want to feel myself just like a noble gas, I will count your shared electrons as if they are mine. And whenever you want to show or feel um, like a noble gas, you count my shared electrons as if they are just your own. So this is there. So oxygen needed 2 electrons. So over here, between the carbon and oxygen, 2 electrons are shared. 2 electrons are shared, right? So let's see what happens with the number of electrons. So now suppose that this oxygen wants to feel like a noble gas and wants to show off that it has the electronic configuration of neon, which is 2,8. So let's count it. 1, 2, 3, 4, 5, 6. Now it will count the shared electrons of carbon as if they are its own. So 1, 2, 3, 4, 5, 6, 7, 8. So finally the oxygen has 8 electrons around it. Now oxygen is happy because it feels, it has not, but it feels as if it has electronic configuration that of neon. Now, let's come to the carbon. Now, we need to count the number of electrons around carbon. 1, 2, 3, 4, 5, 6. So it has still 6 electrons around it. So it cracks another deal with another oxygen atom with 6 electrons in the valence shell. And out of those 6 electrons, 2 form the bond with the carbon. And let's see, does the second oxygen can feel like a noble gas? Let's see. 1, 2, 3, 4, 5, 6, 7, 8. Yes, now it can feel like. And what about carbon? Let's see the number of shared electrons uh, around the carbon. 1, 2, 3, 4, 5, 6, 7, 8. So we count all the electrons in the valence shell along with the shared electrons, right? So over here, so this is the main thing, the structure. Just analyze. So carbon is forming 4 bonds in total, uh, out of which, so actually there are 2 bonds only. Those 2 bonds are double bonds, but when we really want to count, there are 4 bonds. 1, 2, 3, 4, right? 2 sigma and 2 pi bonds. So we can see now carbon needs to form 4 bonds. So this gives us another term which is called tetravalency. So carbon shows tetravalency. Carbon shows tetravalency. Now, what is this tetravalency? Tetravalency, if I actually 4 valency means carbon has valency of 4. Now, what does that mean? So for that, we need to first understand the meaning of valency. Now, what is valency? Valency is ability of any atom to take or give electrons. Like, and the number of electrons it can either take or donate or give is called valency, or is the valency of that particular atom. Now, for example, over here we can see that carbon has the ability to gain 4 electrons to become like neon, right? Therefore, its valency is 4, and that's why it is called tetravalency. Now, it is not always of just ability to have the electrons. It is it is also ability to give the electrons. For example, lithium, it has atomic number 3. Its electronic configuration will be 2 1. And now we would say that okay, in the second shell there must be 8 electrons, so it needs 7 more electrons, so its valency is a 7. No, its valency is not 7. The nearest noble gas to lithium is basically helium with atomic number 2, and the electronic configuration just 2 electrons in the first shell, right? So it will not look up to neon, it will look up to helium, and it will here love to lose this 1 electron, love to lose this 1 electron, and it will become lithium plus, right? So it has ability to lose the electrons over here. So, and it's because it is losing 1 electrons, therefore its valency is 1. So valency is, it is the ability to lose or gain the electrons to have the electronic configuration same as that of nearest noble gas. Carbon is tetravalent, which means it shows tetravalency, which actually means that carbon will, now we will go through whole of this biochemistry. You will see so many structures of the biomolecules. You will always see that carbon is forming 4 bonds around itself. It can be 2 double bonds, it can be 4 single bonds, it can be like this as well, like three, 1 triple bond and 1 single bond. It can be like anything like that, right? So this, we will have to always satisfy its tetravalency. Whenever we will be drawing the structure, any structure, we will have to look for that if the tetravalency of the carbon is satisfied or not. So this was the way, this we have discussed the carbon in detail basically, because this is again the basis of all the biomolecules. This is the atom which is the basis of all the biomolecules basically. So now this is this kind of structure we need to see. Either of these 3 things we need to witness, right? If it is not witnessed, then it means tetravalency of carbon is not satisfied, and certainly our structure is not good to go. Another thing about carbon is that carbon undergoes catenation. We have discussed that carbon has the tetravalency. Another important thing about carbon is that it shows catenation. It shows catenation, which means that carbon has the ability to form the carbon chains, which means that one carbon atom loves to form a covalent bond with another carbon atom. That another carbon atom loves to form a covalent bond with another carbon atom. This another with another, this another with another. So there are always, you would see so many carbon chains when we will be doing the biomolecules. Like it's not restricted to carbohydrates. There are all these things which we have done. This all this discussion is not just meant for carbohydrates. No, this will be these things will be useful throughout the biochemistry basically. Now, challenges, that how to identify the saccharides, like whether that is a carbohydrate or not, how to identify that. The best thing is that we should know the saccharides chemically, that chemically what they are, right? They are, saccharides are polyhydroxyaldehydes and polyhydroxyketones. Does that seem complex chemical definition for you? If it is not, very good. If it is, then I will make it simpler. So for that, I will have to explain you few things related to organic chemistry. So as we have discussed that carbon forms the carbon chains, right? So let's form one such chain over here. Carbon binding with another carbon binding with another carbon, and let's take this chain of just four carbons. Now, second thing which we have learned is that carbon is tetravalent, which means it needs to have four bonds around it. So let's see, we need to fulfill its valency, right? So this carbon over here has just one bond. So it needs three more bonds around it. It needs to form three more bonds with three more species, right, atoms. This carbon has two bonds already, so it needs two more to make it four. This carbon has two bonds, it needs two more. This carbon has one bond only, so it needs three more bonds. Now, yet this tetravalency is just satisfied by the hydrogens, just by all the hydrogens over here, right? It's the ones, the atoms which are satisfying its tetravalency for all these carbon atoms are the hydrogens. Then this is very basic hydrocarbon, which are generally, it belongs to the class alkanes. So what are alkanes? Alkanes are the hydrocarbons, like hydrocarbons are what? Are the carbon compounds where you can see hydrogens and carbons, right? So many hydrogens and carbons. So these are basically the alkanes. And what is the property of that? That there is a chain of carbons, and that chain is made by just single bonds, and all the valencies of all the carbons are satisfied just by the hydrogens. Then it will become alkane. Now, uh, because there are four carbons over here, so which type of alkane it is? It is butane. It is butane. For the four carbon chain, the prefix name is but. One, two, three, four. It is but for one carbon compound. It is meth. For two carbon, it is eth. Three prop. Four but. Five pent. Six hex, seven hept, eight oct, nine non, and 10 dec, like that. Right? So over here we have just, this is alkane because there are all the single bonds among the carbons, and the valencies of all these carbons are fulfilled by just the hydrogens. That's why this is alkane. So, and it is butane. Now, any of the two terminal carbons. So let's say this one decides to form a bond, wants to satisfy its valency not by hydrogens, but with the help of oxygen. Now, as we know, the oxygen tells that if you really want to form the bond, then you will have to form the double bond. So the oxygen forms the double bond over here. Now, because the oxygen forms double bond over here, then let's count the number of bonds around this carbon over here. One, two, three, four, five. So there are five bonds right now, which is not possible, right? It has just a valency four. So this hydrogen also needs to be substituted, right? So for two hydrogens are substituted by a double bond oxygen. Now, this group over here, which is just bonded, like this is carbon double bond O. This group over here is called carbonyl group. This group is called carbonyl group. Carbonyl group. Now, if this kind of situation arises that either of the two terminal carbons, if they decide to just fulfill its their valencies with oxygen, a double bond oxygen, then this the entity of this biomolecule changes and it becomes aldehyde. It from alkane, it will no longer remain alkane, it will become aldehyde. This becomes aldehyde. Now, similarly, let's make another chain, which is just made up of, again, made up of four carbons, right? They're all their valencies satisfied by the hydrogens. So we are again starting the thing from the very basic hydrocarbon, which is alkane. Now, this time, instead of the terminal carbons, the central carbons decide to go with oxygen, right? So for that, certainly, if these are the central carbons, right? To go with the oxygen, one of this hydrogen needs to be just replaced, right? So over here, there a double bond will be formed and this oxygen will be formed. Now, if you count the number of bonds around the carbon over here, 1, 2, 3, 4, 5. So there are total 5 bonds over here, which is not possible. So this hydrogen over here will also be eliminated, or we can say substituted, or we can say replaced by the double bond O. Now, if you will see, this is again a carbonyl group. Again, this is a carbonyl group. This is C double bond O. This is again C double bond O, another carbonyl group. But this time, this carbonyl group is in the center of the, or in the middle of the chain. And if this kind of scenario arises where the carbonyl group is in the middle of the chain, then in that case, this group is called ketone, right? So this group will be called ketone, right? So you can see over here, there is a carbon, terminal carbon with a carbonyl group. Two of its valencies are satisfied by oxygen. One is satisfied by another carbon over here, and one of them is satisfied with the hydrogen. While when we come to the central carbon, when it satisfies it satisfies two of its valencies with oxygen, and two of them are satisfied by the two carbons on both the sides. So these are called ketones and these are called aldehydes. Now, forget about these carbons with the carbonyl group. Let's now go to the other carbons in the same molecule. For example, all the carbons like 1, 2, 3, rest of the three carbons, they also decide to form the bond with oxygen, but the oxygen which they find, it is already associated with one hydrogen. So here oxygen says, because I have already formed one bond with a hydrogen over there, so I can form just a single bond with you. So says, okay, this carbon also forms OH bond with O and H. This also forms OH chain. Now this OH group over here, this group is basically the alcoholic group, OH group. And this group is also called hydroxyl group. So this group is also called hydroxyl group. We go over here. This is hydroxyl group. So, and if it is there in any of the biomolecules, it means it is giving the alcoholic character to that biomolecule. So now, if you will say this is aldehyde, right? One thing is very clear that this is aldehyde. Now, there are so many hydroxyl groups in this aldehyde. That's why, because there are more than one hydroxyl groups, each and every rest, apart from the carbonyl group, each and every other carbon in that chain is satisfying its valency, one of its valencies with the help of hydroxyl group. That's why this molecule now will be called polyhydroxyaldehyde, right? Now, if same thing happens over here, like this is the carbonyl group carbon, but apart from that carbons, all other carbons just replace these hydrogens with OH, with OH, right? It also forms with bond with OH. This also forms bond with OH. This also forms bond with OH. Then in that case, it will become, suddenly there are so many OH, so it will become polyhydroxy ketone. It will become polyhydroxy means many hydroxy, ketone with many hydroxyl groups, ketone with many hydroxyl groups, right? Polyhydroxy ketone, right? So I hope now the chemical definition of saccharides is clear to you. So saccharides are either polyhydroxyaldehydes, means either they have the carbonyl group at the terminal carbon, right? With rest all the carbons having the hydroxyl groups. So they are either the polyhydroxyaldehydes or they are polyhydroxy ketones, which means that within the carbon chain, one of the carbon is forming a double bond with oxygen, forming a carbonyl group, while rest all the carbons in the molecule are getting associated or forming bond with the hydroxyl group. So this is polyhydroxy ketone. So same thing over here. Here I have drawn the horizontal diagrams. Same thing is shown on the left hand side with the help of vertical diagrams. Like they are the both the things are very similar. You can see this is the molecule with the three carbons, right? We did it with four carbons. This is molecule with three carbons, and this is the terminal carbon with the carbonyl group. So therefore it is aldehyde. This shows that it is aldehyde, right? Okay, but this aldehyde is having so many hydroxyl groups also associated with the rest of the carbons, and therefore this is polyhydroxyaldehyde. Polyhydroxyaldehyde. Similarly, over here, this is again a three carbon compound, and over here this is the carbon with the carbonyl group is middle one, while rest of the carbons fulfill one of it, the valencies with hydroxyl groups. So there are two hydroxyl groups. So therefore, this is polyhydroxy ketone. Polyhydroxy ketone. So if we see any molecule satisfying these conditions, then certainly that one is saccharide. If it is polyhydroxyaldehyde, those polyhydroxy aldehydes, those saccharides or those monosaccharides are called aldoses. And if they are polyhydroxy ketones, they are categorized as ketoses. In the next session, we can just discuss uh, the structures of different aldoses and ketoses in detail. Till then, goodbye. See you next time.